The human eye can see in both colour and in black and white. Colour sensitivity is useful for identifying ripe fruit or spotting certain forms of camouflage. However, for colour vision to work well, it needs a large amount of light in order for the eyes to distinguish between different wavelengths of light. So in low levels of light, the eyes rely upon the black and white vision. It does mean in dim light some of the nuances of colour vision are lost, and even sharp focus on some objects is also lost. However, items that can't be seen in low light to the colour vision are still able to be perceived using the black and white vision. The general principle here is that rod cells in the retina of the eye are the site of the photoreceptors for black and white conditions, and the cone cells are the site of the receptors for colour vision. A smaller number of the cone cells are most closely gathered around the focal point where the light hits the retina or the foveae centralis, the back of the eye, allowing for good colour vision on whatever the eye is actually concentrating on. And the rod cells, which outnumber the cone cells by about 15 to 1, gathered around the outer edge of the retina. They can produce some of your peripheral vision. Now, as well as the rod cells being far more numerous than the cone cells, they're also far more sensitive to light. Even a single photon is enough to stimulate a reaction in a rod cell, whereas maybe 100 photons or more may be required to stimulate a cone cell. In addition to this, the rod cells are thinner, so can be more densely packed than the cone cells. It does leave a question. If there are more of the rod cells and they're more sensitive, why then isn't your daytime vision dominated by the signals coming from the rod cells? Well, this relates to what happens once the signal's actually been received, and the structures in place for passing on that reception. The rod cells operate in groups. The message from the group of the cells is transmitted down a single bipolar cell. This means that even if all the rods are stimulated at the same time, only one signal can be transmitted at a time. Cone cells, however, each have a dedicated bipolar cell, which only transmits the signal from that cone cell. In bright light conditions, this will result in the signal from the rod cells being kind of lost in the signals coming from the cone cells. Additionally, in bright light, there's nearly always going to be enough light to stimulate the rod cells. So in order to prevent the brain from being overloaded with light signals from the rod cells, the sensitivity to these cells is kind of turned down in bright light conditions. Now, by having a dedicated cell response for the transmission of the signal from a cone cell, also means that the image produced can be sharp and focused, whereas the signal from the rod cells comes from a kind of a generalized area, meaning the resultant image is produced as a poorer resolution. Now, cone cells themselves are split into 3 types, each responding to a different wavelength of light, rather than just a specific wavelength. These cells are normally referred to as S, M, and L, so they respond to shorter wavelengths, medium wavelengths, or longer wavelengths of light. But in reality, they should respond to what might be termed shorter, longer, and very long wavelengths. So there's generally a large gap between the optimum reception of the short and medium wavelengths, corresponding to kind of blue-green or cyan, and then a small gap between the reception of the medium and long wavelengths in the red part of the spectrum. Within the cone cells, there are photoreceptor proteins known as photopsins, which actually respond to different wavelengths of light. The exact range of wavelengths that the cells respond to varies from person to person, as does the specific perception of colour. This can result in an inability to perceive differences between 2 colours, what's known as colour blindness. Some people may even have a 4th type of cone cell, giving them a totally different perception of colour. This may explain some of the more colourful artwork that's occasionally produced by some painters. When light levels reduce, rather than just relying on the previous amount of light entering our eye, the eye adapts by opening up the iris, which is the colour part of the eye, allowing more light to pass through the pupil and so onto the retina. Now, by allowing more light to reach the retina, the eye can make the best use of the available light, enabling small details that would otherwise be missed still to be seen in dim light. So why then doesn't the eye always allow more light into the eye, produce a better picture of the surroundings? Well, each time the protein photoreceptors in the retina react to wavelength of light, They then need to be recharged before they can respond again. By managing the amount of light reaching the receptors, the eye can manage the rate of recharge without having periods of reduced vision or being overwhelmed with signals. Changes in the iris happen naturally and fairly rapidly when they move from one light condition to another. However, if this was the only process occurring within the eye, it wouldn't account for the slower process which also seems to occur. The longer we spend in a fairly dimly lit environment, the more our eyes seem to adjust to the low light conditions. This slower adjustment can't be just put down to alterations in the iris, as just after a few seconds it's already adjusted as much as it's going to. Now, the brain is partially responsible for this slow adaptation. It changes what information it now requires from the eyes to form a picture. It basically now kind of turns up the sensitivity of the signals from the rod cells. However, the brain cannot produce a picture if light isn't actually getting some kind of response from the retina. So something else must be occurring in the rod cells. Now, where cone cells have photopsins as the photoreceptor proteins, in the rod cells they have something called rhodopsin, which corresponds to the light between the short and medium wavelengths of the colour vision. So your black and white vision is basically a monochromatic version of colour purple vision. The amount of this photoreceptor is far greater in the rod cells than the photoreceptor in the cone cells. It plays a significant role in why the rod cells are more sensitive to low light. However, the reaction to the stimulation of the photoreceptor is greater within a rod cell than with a cone cell, again aiding low light vision. In normal light conditions, the photoreceptor proteins in the cells undergoes a chemical change known as bleaching when it interacts with light. It means that the photoreceptor has been converted back again to its unbleached state before it responds to light. In low light conditions, this bleaching process can be kind of shortcutted by the use of retinol, a form of vitamin A. In order to facilitate this shortcutting process, it does take about 10 minutes in the cones and about 20 minutes in the rods to adapt to low light conditions. It means if you go from a brightly lit room to a dark street, it will take about 20 minutes before your eyes are operating at their optimum night vision mode, even though the adjustment of your iris is just completed in a few seconds. Reverse is also the same. Moving from a dark environment to a bright environment, the eyes can be temporarily dazzled by the additional light. Then the eyes take a few minutes to fully adjust to the light level so it no longer feels quite so harsh. Role of vitamin A does mean that whilst eating carrots won't enable you to see perfectly in the dark, a deficiency in vitamin A will mean that your night vision will be impaired. So if you do actually have poor night vision, it could be an indicator that you do have lack of vitamin A in your diet. However, before you try eating loads and loads of carrots or other foods with high in vitamin A, it is important to realise that too much vitamin A can also be toxic. So a balanced diet will normally provide all the vitamin A that you need for both your eyesight and for other healthy body functions.